I never know where to stand. Are we starting? Oh, we've started. Hey, you guys, welcome to the review. Almost spring break. So I'm going to go ahead and start this if you all are in that want to be in. How many slides are in this slide deck? Yeah, 7. There's no way we're getting through 70 here in this review. This was the same. Your hair looks so pretty. This is the same slide deck that you had in class. So I want you to think through what were the things that were most, we were worried. What are the things that were most concerning to you or that you didn't get to? Remember in class, I also gave you a link to all the quizzes that we did, right? So you can do those again. So when we look, I didn't do a single list. I kind of broke the list up, but you got to remember we have the circulatory system, we have homeostasis, then we had the circulatory system, we had the immune system, we had the endocrine system, and we had the nervous system, right? So if you reflect back on what is worrisome to you, can you just verbalize that with the person that you're sitting next to? Like, hey, I need to make sure we talk about meh, whatever that might be. It's just a minute.
It's probably the heart of just keeping right mind for left and right streets.
Okay. Can I get some shout outs? I'm literally going to write them down here so I make sure I go over whatever you want me to go over. Nervous system. Okay. Are you going to tell me all the systems? So nervous and lymphatic system are your biggest concerns? Yes. Yep. Heart and the hydrostatic versus osmotic. Okay. That's at the capillaries. Got it. What else? Pardon?
Parts of the brain.
Parts of the brain. You got it. Anything else? Yes, I'm sorry.
The different types of like attack cells, like the police and like this.
You got it. That'll be lymphatic. Okay, types of cells. Anything else? Should we start with that and then see where we get to? So that says to me that you're pretty good about homeostasis, right? So just reminding you on homeostasis, the difference between negative feedback and positive feedback. What I would remind you is negative feedback is more gets you less, and positive feedback is more gets you more, right? And which one do we have more of in our bodies? Negative feedback, exactly. Both of them are trying to maintain and bring us back to homeostasis. Good. All right. So unit 7, we talked about the structure and function of blood vessels, the heart, chambers, valves, the blood pathways, components, and disorders. So what I heard from you just now, that you were just kind of mostly concerned about the capillaries. Do you want to do the song one time, or you got that down? Here we go. Arteries go away from the heart, veins go towards. We're going to remember that, right? Because you know I'm going to ask you a question about that. Okay, do that again. Arteries go away from the heart, veins go towards. Arteries, arterioles, capillaries in the middle, venules, veins. Heart, body, heart, lungs, heart, systemic, heart, pulmonary, atria collect, and ventricles pump. Then what we need to remember is words like pulmonary has to do with lungs, coronary has to do with heart, hepatic has to do, did I teach you that? Yes, I did. Hepatic portal vein, remember that? The junction between the stomach, small intestines, and your liver. Remember that's a place where you have double capillaries? So sounding familiar now? Okay. Hepatic is liver, renal is kidney. Or something else there. Oh, you need to keep your lefts and rights straight. Yeah. So when you're looking at a person, right, when you're looking, if there's a drawing on here, it's their left, right, and their right. And remember, left starts with the letter L. Your lungs start with the letter L. So it's the left side of your heart that has oxygenated blood. The right side of your heart has deoxygenated blood. And I'm bound to ask you some sort of question like, which chamber of the heart collects blood, which chamber of the heart pumps blood, right? So be prepared for that. Okay, so you asked about the capillaries, so let me get there. Hopefully I have a slide there. If I don't, I'll come back to it. You know that. Structures of the heart. Good on that, yeah? Okay. Hopefully do I not have a capillary picture? Oh, you know, pulse is a measurement, right, of how fast your heart is beating, that expansion of the arterial walls. Blood pressure is when you have the greatest pressure is your systolic, when your ventricles are contracting. What do you call it when they're relaxing? Diastolic. We're going to remember that because if they relax too much, you will. Okay, good. Okay, we did pulmonary, systemic. Oh, here, that's what you're talking about. Okay, when you come upon a capillary in your systemic circuit, right? Arteries go away from the heart. So the it goes arteries, arterioles, capillaries in the middle, right? At the arterial end of the capillaries, the blood pressure is pretty high. And then as it gets farther away from the heart and starts to make its return trip in the capillary bed, the blood pressure starts to drop. So when we look at a capillary bed, the pressure, the hydrostatic pressure is greatest at the arterial end, and then it kind of fades away. Okay, still there, but okay. The osmotic pressure is pretty constant throughout the whole thing. Remember, osmotic pressure is saying come in, and you can think water must. So it's flowing in because, you know, there's all those things in blood plasma. Remember road trip, right? All those things in blood plasma. So fluids want to come in. But at the arterial end, the blood pressure is greater than the osmotic pressure. So it pushes things out like oxygen, like nutrients, right? And then by the time you get to the venular end, then the osmotic pressure coming in is greater than the hydrostatic pressure because the hydrostatic pressure has fallen over the course of the capillary bed. And so you're recovering CO2 and you're recovering waste. That's why we say all exchange takes place at the capillary. It is one cell thick. That's the only place where it's one cell thick. So the hydrostatic pressure can have its impact and the osmotic pressure can have its impact. Does that help you? You need me to go over anything more on that, you guys? Okay. All right. So we talked about plasma. All right. So now going into the immune system, you all said lymphatic system, and specifically I did that. Okay. So I specifically see types of cells. Okay. So I'll hit that. Non-specific defenses, specific defenses, and then in getting that immune response to happen and any side effects where it was the overall. Remember, we talked about more gets pushed out at your capillary bed, more fluids out than gets recovered. And then that tissue is called interstitial tissue or tissue fluid. And it's going to move into your lymphatic vessels. And then it's called what? Once it moves into the lymphatic vessels? Lymph. And before that lymph is returned to your circulatory system at your subclavian vein, it's going to pass through some lymph nodes. So your lymph nodes filter your tissue fluid. And that's where those guys we're going to talk about like to hang out, because that's a good place. All fluid will pass through the lymph node. Blood, where is blood filtered? Spleen. Good job. Okay. You also absorb fat at the intestines, and then you produce those lymphocytes. So we already talked about that. Remember, listen to me, all blood cells come from the bone marrow, but B cells mature in the bone marrow, whereas T cells mature in the thymus. You're going to remember that? Okay. And we already talked about these two things right here. All right, so now let's talk about these different cells. So red blood cells, you know what red blood cells are going to do? They're going to carry oxygen. Platelets are going to help your blood to clot. Good. And then when you had your granulocytes right here, what is the most numerous of those? Which ones? Neutrophils, most numerous. First on the scene. Who's the big eater? Macrophages are the big eaters. Who's the thing that secretes histamine? Mast cells. Good. Okay, good. On your innate immune response, if you know that, you're set. Okay. Of your special forces, remember the natural killer cell. Remember he dropped out. He was supposed to go to the school of thymus. Okay, so he is a lymphocyte, but he just likes to kill. He's part of your innate immune response, meaning you are born with it, right? This right here, your B lymphocytes, T lymphocytes, these are your specialists. The T lymphocytes and the B lymphocytes, you might have one or two that can recognize a specific antigen, but once they're triggered, and we could talk about that triggering process, once they're triggered, they will make a clone army, millions of cells who can fight that one specific antigen. And when the battle is over, they have to commit what? Yeah, because we can't maintain those high numbers. The only thing we leave behind for B and T cells are to make it happen faster. Perfect. Faster. Yes.
How does it happen faster if it's still like there's just
To
one
trigger
cell?
it is a lot easier than a virgin cell. A virgin cell, remember how the B cell has to come in contact with the specific antigen, and it has to come in contact with the T cell who has also been stimulated, and they have to agree, yes, before they start. Non-virgins aren't like that. They come in contact, boom, they're ready to go. Yes.
What secretes the cytokine?
Cytokines. Okay, cytokines are good. So you have several. That's just any kind of chemical. We talked about interleukin and interferon, right? So macrophages can secrete that. It's like pulling a fire alarm and saying, hey, we have problems here. So it alerts the rest of the immune system. That's interleukin. Okay, interferon can be secreted by a cell that's infected. And so it's trying to warn everybody else, hey, if you're anywhere near me, you should stop doing protein synthesis and you should probably just kill yourself because you're probably infected. All right. Now, okay. First things first, remember, we'd rather just keep things out of our body. So that's skin's job and saliva and low pH of our stomach. But if it does get in past those, what's going to trigger any kind of immune response? The antigens are on the pathogen, right? What is it called if we think our own cells are carrying antigens? Autoimmune disease. Good. All right. So here again shows your please. So let's take a look at those and make sure we're good with it. What is this? Okay. So macrophages eat. They started off as monocytes to get out smaller, and then they became huge. Dendritic cells, they're eating and displaying. They're called an antigen presenting cell. Who are they going to need to display to? A B or a T? T cell. Remember, T cells do cell to cell combat. Maybe you could think of it as T like this and punch, 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 punch. They do cell to cell combat. And so they need to be stimulated by one of our own body cells displaying that antigen, either an infected cell displaying that antigen, and they would display on an MHC 1 and you're done. Or if you are an antigen presenting cell, like a dendritic cell or a macrophage, you would display on an MHC 2 and I will help you. Perfect. Okay. Natural killer cells. We got, complement proteins are part of your innate response. And what we learn mostly about that is they like to drill what? Holes. They like to drill holes to kill. Neutrophils most numerous. And then we have our B and T cells. So this is a good summary of those as well. Okay. Okay. All right. Inflammatory response. The inflammatory response says your innate immune response is working. Okay. That swelling, that localized heat, the tenderness means that fighters are there. Histamine is coming from the mast cells, making the blood vessels leaky. So it makes it easier for fighters to get there, elevate the temperature. You're trying to kill off any bacteria that are there, right? So those are all good signs that that infection is getting taken care of. But will that do it? No, we have to enlist the special forces if we really want to wipe everything out. And this is just another slide kind of walking you through the inflammatory response. So comparing and contrasting, cell-mediated is T cells. Remember, what do? Cell to cell combat. B cells will eventually make what for you? Antibodies. And antibodies can flood your body fluids and mark anything. They're very specific, right? Very specific. So there's one pathogen that they'll tag, one antigen that they'll tag. A different B cell has to take care of a different one, right? But when they make those antibodies, they're specific to that antigen and it'll tag it. It'll tag it to be eaten by a macrophage, or it'll make it so a virus cannot get inside your body cells. It kind of deactivates that virus. two different ways. These antibodies take care of those pathogens out and about in our body, in our fluids, whether it's our lymphatic fluid or our tissue fluid. Okay. The T cells are cell mediated. That takes care of our own cells that are harboring those pathogens. So we need to kill those cells off so they don't release more viruses or more infection, right? So the only way to talk to a T cell, remember MHC1, if you display an antigen on MHC2, I will help you. Good. So we talked about that. This is just another Venn diagram that says the same thing. Remember, it's when the virgin B cell is stimulated, it makes plasma cells that are releasing those antibodies. And what's the other cell it makes? Memory cells. And the way you're going to remember that is the plasma cells are releasing the antibodies out into your blood plasma, right? And if it's out in your blood plasma, it's going to get out into your tissues too, right? Okay. And then T cells also make memory cells, and they make helper T cells and cytotoxic T cells. Cytotoxic T cells are the ones that are going to do the killing. Okay. Helper T cells are going to do the stimulating, stimulating the B cells, et cetera. They're like, let's go, let's fight. Okay. All right. Now, as far as your immunity, what you're born with, and then what you can acquire, either active immunity because you've been exposed to the pathogen directly, or you got a what? Vaccine, or you can borrow immunity. Borrow immunity from your mother when you were a baby, or you can borrow immunity through a shot, a gamma globulin shot. Okay, that's it. Let's see. So now that moves us on. We have the nervous system, hypothalamus, and the endocrine system. Okay, I think we're good on that. I'm looking. Okay, good. So compare and contrast the nervous and the endocrine system, protein versus steroid hormones, positive versus negative feedback, and contrary hormones. We've talked about that. Glands, location, hormone function. Did you see that last slide I put here in this review deck where I said, hey, you should know how to do this? Did you guys all look at the last slide in this slide deck? That's a good slide. It's a really good slide. As a group, I would look at that slide. As a group, I'm just saying that I would look at it. Okay, exocrine versus endocrine glands. Endocrine glands secrete into your what? Blood. Exocrine glands are like sweat glands or salivary glands. They're going into ducts, right? Okay. How do endocrine glands work? They work by, they get released everywhere, but only certain cells have receptors. Good. Okay. Comparing and contrasting nervous to endocrine, I think you already know which one's faster, which one's effects last longer, which one uses negative feedback, both. Good. Which one uses a neurotransmitter? What uses a hormone? They're both chemicals moving at a distance. Which one moves at a greater distance? Endocrine, right? Because it could be secreted by your pituitary but affect your thyroid, right? Whereas neurotransmitter are just having to cross the synaptic cleft. Remember that? Synaptic cleft, where two neurons meet, a synapse. Does that sound familiar now? Okay. We already talked about feedback, so I'm not going to hit it again. Contrary hormones like insulin and glucagon, I would know that. That's such like a ubiquitous one. I would know it. Insulin's going to do what to your blood sugar? And glucagon's going to raise it. And they're doing opposite, right? You know what's another one that does that? Calcitonin from your thyroid gland. Calcitone it down now. It lowers blood calcium. What's the one that raises it? Parathyroid gland. Perfect. Those are good. If you know those, you're set. We already talked about positive feedback, negative feedback, hypersecretion, hyposecretion. Hyper is too much, hypo is too low. Here's what you need to know about these two guys. Okay. Protein hormones cannot enter the cell because they can't get through the phospholipid bilayer, right? So they stand at the door and they trigger some secondary messenger inside. Cyclic AMP, open some gate, something. They don't get to come in. Steroid hormones, because steroids are a type of lipid, they can go through the phospholipid bilayer. So if you do something to alter the cell membrane, which hormone's going to be impacted? The steroid hormones, because they're supposed to go through it. Protein hormones will be fine because they never cross the membrane, right? All they have to bind to is a receptor. See the difference between those two? Yes? Okay. Oh, and steroid hormones, that has to do usually with reproduction and long-term stress. Okay, stress and reproduction are those types of hormones, very long-lasting effects. Okay, so one of the things on the list here is the hypothalamus, the pituitary gland. So let's talk about that. Okay, hypothalamus, full-on brain. Okay, pituitary gland, full-on endocrine system. Okay, this, remember, your nervous and endocrine system are the two systems that help you maintain homeostasis. This is where they cross over. Okay, now your posterior pituitary, your post-pit, descended out of the hypothalamus, direct connection. Okay, direct connection, hypothalamus, posterior pituitary. So direct, in fact, that the hypothalamus makes the posterior pituitary's hormones. What are those two hormones? They both have to deal with fluid. Oxytocin and antidiuretic hormone. Antidiuretic hormone, does it make you pee more or pee less? So you don't die, right? You don't want to become dehydrated. Oxytocin makes the baby come out and the milk come out. Okay, who makes the milk? Prolactin, and that's in the, okay, hypothalamus, direct connection, ADH, oxytocin. Anterior pituitary, not direct, because the anterior pituitary came up out of your mouth. And so the hypothalamus only reaches as far as the stalk, right? Which is where the anterior pituitary globbed on. And so it puts releasing hormones into the capillaries there, which then get carried by the blood and come out at the anterior pituitary. And those releasing hormones tell the anterior pituitary, release this, release this, release this. And the anterior pituitary says, okay. And what it secretes are what's called what kind of hormones? Stimulating hormones. Good. They're going to stimulate some other gland to do its business. So the hypothalamus bosses the anterior pituitary, and the anterior pituitary bosses everybody else. And that's why the pituitary gland is called the master gland. It's like the gland. Yes.
So does the anterior pituitary make the?
Yes. Anterior pituitary makes its own hormones. It's just that the hypothalamus tells it what to release and when to release it. Yes.
So would it mean that the posterior pituitary would like release the hormones faster since it's directly connected and the anterior has like?
Well, that's a really good question. So probably a little bit faster than the anterior pituitary, but because of the double capillaries, that solves the problem with the anterior pituitary. That's a great question. That's why that's one of those places where you have double capillaries, is to speed up that hormone to come from the anterior pituitary. Okay, what else? Good on that? Okay, so I'm crossing that off my list. Okay, so I have mostly just nervous system left. So you can review what glands, what hormones, right? And did you see a question I posed for you at the end of the slide deck about some glands? Okay, good. Adrenal cortex, long-term stress, adrenal medulla, epinephrine, norepinephrine. That is fight or flight. Good. Okay. I think you'll get the testes, ovaries, thymus. You already know that. T cells, pineal gland secretes what? Melatonin for sleep. Okay. Then adipose tissue says it's leptin. Leap away from the table. You're full. It's telling you you're full. You have EPO, which is from your kidneys, and it's telling your bones, hey, make more red blood cells. And we talked about prostaglandins being local chemical mediators. All right, pheromones versus hormones. Hormones travel at a distance within your body. Pheromones travel at a distance between two. Perfect. Okay, nervous system. Break down all the divisions and the functions and know what neurons do. So let's look at that. We know we have sensory neurons go towards the what? Central nervous system. The central nervous system is made of what two things? Brain and spinal cord. And what kind of neurons are there? Inner neurons. Okay, so once they decide what to do, they send that message out to your effectors. What kind of neuron do you use to send it out? Motor. They're motorin, right? They're going to send it out. What are the two kinds of effectors you have? Muscles, and they will just contract, and glands will secrete. All right with that? Okay. So then when we look at the parts of the neuron itself, right? There's an indoor and an outdoor in a neuron, right? The end receiving door, those are called what? Dendrites. Okay. You got your nucleus there, maybe processing occurs. Then you have the long axon. Axon goes away. It ends in these little axonal terminals, or synaptic knobs is another name for those. Here I've got a fatty layer called a myelin sheath, and that's made by Schwann cells. That's good because what's it doing for the neuron? Speeds up the whole thing, and it also what's it? Insulates it. Okay, all right with that? Okay. All right, so now resting potential. And that was on our list here. Okay. For a neuron to work, like for your phone to work, your phone has to be what? Charged. This is how you charge a neuron. Okay, this is how you charge a neuron. The biggest thing you can do to charge your neuron is use your sodium potassium pump. It's going to pump 3 sodiums out for 2 potassiums in. Is that going to cost you energy to concentrate an ion on one side of a membrane? Yes. 30% of your ATP. Yeah. Okay. So that's your biggest thing. Sodium potassium pump. More positives out than in. That right there should always remind you, if 3 sodiums go out and 2 potassiums in, that the outside is going to be what charge? Positive, and the inside is going to be what else can make the inside negative? What do we have inside? Negatively charged ions. That's also going to contribute to it. And also one of our gates is leaky. Okay, which gate is leaky? A leaky gate. Potassium. The potassium gate is leaky. It's like it's kicking. It's like open up gate. Okay, K. Leaky. Okay, and so what happens is, you're like, what's the connection there? I'm trying to think it was like a K. See, it's like kicking the gate open. It's not working for you. That's okay. So the potassium is going to leak out from the inside of the neuron where it's supposed to be, because the sodium potassium pump put it there, right? Now that's all going to help you. And then it's going to leak back outside. So the outside is positive, the inside is negative. That's my phone being charged. Okay. And action potential is just a reversal of charge. It's just like setting off dominoes along the length of the neuron. To do that, who wants to come in? So if a positive charge was outside and now it comes inside, instead of being positive out, it's going to be positive, which triggers the next gate to come open. And now more comes in, more comes in, more comes in. Now I have a reversal of charge along the whole length of the neuron. That's called being depolarized. So resting potential, this is how I set it up. When this reversal of charge occurs along the length of the neuron, that's an action potential. I got to fix it. Repolarization is my fix. What's my quick fix? Potassium. Yeah, all those potassium gates, those leaky gates, they all open. So now it's a positive charge. It takes sodium's place outside and it resets it. That's our quick fix. But our long-term fix, we better put everything back where it's supposed to be. So sodium, you rushed in. I'm going to put you back out. Potassium, you repolarized, you fixed. I'm going to put you back in. I'm going to clean up the mess. Okay? The sodium potassium repolarizes. So let's think that through. Resting potential. What 3 things contribute to the resting potential? Number 1, sodium potassium pump. Number 2, negatively charged ions inside. Number 3, leaky potassium gates. That's my phone is charged. Okay? That's my resting potential. Action potential is a quick reversal of charge that occurs because sodium rushes in and it makes, remember on that graph, it makes it positive inside. How do we get it to be negative on the inside? What goes back out to fix it, to repolarize it? Potassium goes out. Good. Okay, and that's what this graph is right here. Resting potential is negative inside because it's positive outside. Depolarization, sodium comes in. Quick fix, repolarization, potassium goes out, and it overcorrects to keep the flow going in one direction. Okay, that is the electrical part down the length of the neuron. Once we get to the end of the neuron, now it's going to get chemical. And so what's going to happen is when the action potential reaches the end of the neuron, we have a new gate that opens up. Does anybody know what that new gate is? It's not on this picture. Calcium. Yeah. Calcium starts to come in, which makes these synaptic vesicles fuse here with this Pre comes first, presynaptic membrane. Release your neurotransmitter into the cleft, and it'll go to the postsynaptic neuron and bind to a receptor. And if it's excitatory, it'll trigger another action potential. If it's inhibitory, it won't. It doesn't. Okay. And you can see the steps of that: calcium coming in, synaptic vesicles fuse, releasing of neurotransmitter, binding to receptor on postsynaptic neuron. Remember what we said? Drugs interfere with synapse, right? Drugs either facilitate or inhibit a neurotransmitter from being released, or they don't allow it to be broken down or reabsorbed, or they mimic a neurotransmitter. That's how drugs work. Okay. Now, the whole big picture. You have your central nervous system, which is your what? Brain and spinal cord. I'll keep going. Did the bell ring? No. Okay. Brain and spinal cord. Then everything outside of your brain and spinal cord is your peripheral nervous system. So you have nerves coming off your brain and you have nerves coming off your spinal cord. When you break those down, some are sensory going towards, some are motor going away. Going away, we talked about somatic versus autonomic. What am I moving right here? Somatic. This is my choice. Okay? So this is my choice. Autonomic sounds like what word? Automatic. So those are things you don't have to think about, like your heart beating or breathing or anything. That's autonomic. Now, sometimes I want my heart to beat faster. If I'm going to go run in a track meet, then I would need which division of my autonomic nervous system? Sympathetic, because I have sympathy for somebody who's under stress, fight or flight. Yes? If I'm resting, to rest and digest, that would be my parasympathetic. Okay? So a reflex arc, you touch a hot pan, sensory neuron, synapse in my spinal cord to a motor neuron. I pull away before I even know that it's hot, right? That hotness comes later. So a reflex arc is very protective. It's a quick, immediate response when we're in danger. Your autonomic nervous system are basically internal reflexes, always regulating your heartbeat, your breathing rate for you. You don't even have to think about it. You don't have to process it. Okay, this is that broken down. Does that chart make sense to you? Okay, what we already talked about. Okay, ideas to study. Oh, parts of the brain. Let's talk about that. For some reason, I didn't put a picture of parts of the brain. Okay, memory, learning, thought. Cerebrum, right? My cerebrum is memory, learning, thought. Occipital lobe is about here. That's about my vision. What's right here that helps me with my balance? Heartbeat, breathing rate, starts with an M. Medulla oblongata. Those are the parts you need to know. And then I would know these right here. Okay, you did it. Good job. Okay. Is there anything more? No.